So it's a great pleasure for me to welcome Simon Collins from the UK Community Advisory Board and from iBase in London, who's going to talk about a community perspective on PrEP. Simon.
Thank you, Andrew, and I'd like to thank the conference organisers for the opportunity to bring a community perspective to PrEP. I haven't put a disclosure slide in, but I have no financial disclosures. As it's a community forum, I'd like to start with a few pictures of some of my friends from the 80s, late 80s and early 90s who weren't able to benefit from treatment or from PrEP. And I put it in really just to try and reflect and remember what the times were like back then. I think if PrEP had been available, it would have fulfilled criteria for a rapid access program that would have been cost effective and affordable. We would have had so much use by now that we would wonder why we're even having these discussions. Okay. So the previous slide was that it's a community forum, so I wanted to include some of my friends from the 80s and the 90s who weren't able to benefit from treatment. And the context is that if PrEP had been available at this period, I don't think we would be having these discussions about whether or not we should use it. In preparing the paper, I was surprised to find that the first efficacy data for tenofovir was published in 1995. And this was a year-long study, so the research had been going on in 1994, maybe 1993. And the data is pretty impressive, I think. This was pre-heart, it was pre-effective treatment, it was before protease inhibitors, and we had this indication in a macaque study that all animals that were given tenofovir remained uninfected. The study design, it had four active arms, and people were given tenofovir daily, and the researchers had the foresight to think about giving a pre-exposure dose, which is not dissimilar to some of the ways that PrEP is being used today. All the control animals became quickly infected within 1 to 3 weeks. So as part of that timeline, a very brief timeline I guess, is that various independent researchers continue to do macaque studies, and although there's discussions that other ARVs may work and some of the ARVs we have today may work, back then AZT didn't produce very encouraging data. So there may be something specific about tenofovir that has a safety profile and a long half-life that makes it particularly suitable. Throughout the talk, one of the themes I will come back to, I guess, is that all this research has been driven by independent researchers, and it's been driven by a community need for alternatives to condoms. And it's never been an industry priority. Even amongst industry, I think they're quite happy to admit this. I remember being at the retrovirus conference in Seattle, I think it was in 2002. Bill Gates gave one of the plenary lectures, and Dr. Mike Yule stood up from the audience and asked about the opportunity of using ARVs to prevent infection. And the perspective he was doing this from was for an alternative to condoms. It wasn't an alternative to add on to condoms or to make condoms more effective. It was because in the real world, we need better choices that fit people's risks and people's lives. And the response back from Bill Gates was so stunned, it was so rabbit in the headlights, that why would anybody consider having sex without using a condom if they thought they were at risk? And yes, I get there must have had some impact, because Gates went on to found some of the largest randomized PrEP studies. Or the other studies were either privately funded or publicly funded, and yet when Truvada was approved in 2012 with a new indication, there was no adjustment for this public investment in terms of the cost of PrEP. Again from 2003, although gay men and transgender people are disproportionately affected by HIV in many of the epidemics, it's a paper again from Mike Yule highlighted the range of people who could benefit from an intervention that wasn't dependent on the partner knowing that the intervention was being made. So as a background, I guess the holy grail in teams and researchers that are looking at prevention has been an HIV vaccine. And it's always been quite astounding that a vaccine that was 50% effective would be sufficient for use on a population level. And yet on a population level, a 50% chance that you may be protected is not really very helpful at all. We now have pretty convincing data, I think, that PrEP, if you take it, is far more effective than this. As background, I guess, each year maybe 2 million people are newly diagnosed, and that sort of highlights that condom-based prevention programs are continuing to fail a high number of people who are at high risk. An online survey of 1,500 gay men the year after PrEP was approved in the US didn't show a particularly high level of awareness that this scientific breakthrough had now found its way through to the clinic. I think in this study maybe 3% of people had actually used PrEP. So I thought I'd start with a few myths, and one of the myths that's still out there in the community is that this might be a marketing strategy for pharmaceutical companies to make more money, and I think that there's very little evidence to support this. I think that Gilead has done no marketing for PrEP in the direct marketing for PrEP as a company since PrEP has been approved. I think it's something that they've left to public health to take the lead on this. I think Gilead probably have quite a good patient assistance program for US use that's underused, and I think they'd be happier for that to be used more. And I think the potential for really widespread use will come when the cost for this intervention dramatically drops, something like to the level of the price of Viagra or condoms or lube or other affordable things that people use. And so that's clearly not a financial market. A second myth, which is difficult to sustain now, I think, with the PrEP and the EPOGAY studies that have both put out press statements in the last month. But efficacy, I think we've got really good data that PrEP works if you're able to take it. You can't really give a talk about PrEP without referring to some large randomized studies that failed to find a benefit, and that really showed or highlighted the challenges really of identifying people who are really at risk and who continue to be at risk during a study, and also it highlights the issue of adherence. So many of these, in both these studies, I think there were adherence problems. In the last month, both the PROUD and the EPOGAY studies have changed the design of their ongoing PrEP research in order to make the active drug available to all participants, and we clearly need to see the full results in order to understand the implications for use. So I did put in an efficacy slide. This is from almost 5,000 people in a heterosexual study in Kenya and Uganda. 2 active arms, one of them is Truvada, and one of them is tenofovir, and clearly both had a really statistically significant impact on the risk of HIV transmission. There was no statistical difference between the 2 arms. I put in a safety study. 40% of the women in the study were the negative partners, and so although you have to take this safety issues very seriously, there was no alarm really that this is causing a signal for problems, especially compared to placebo. So pharmacokinetics, it's incredibly complex. I'm not going to try and go through the details, but the mechanism of action involves getting active levels of the you've got 2 drugs really with different pharmacokinetic profiles, and you want the active levels of the metabolites in the intracellular levels to be good. There's variability between people, quite considerable interpatient variability. There's differences in tissue absorption between the 2 compounds. And despite this sort of complexity, the efficacy data seems to overcome this on a population level. One of the questions I haven't really seen good data to help me answer is the time it might take to get to steady state, whether you need a different steady state dose in order to achieve these intracellular levels. This is the sort of simple slide I can deal with. So the concept really is that you may need a loading dose, but the target levels for PrEP. This is a slide from Peter Anderson, who works with the IPREX group. It shows that the target levels haven't really been identified yet, and although IPREX suggested that 16 femtomolar may be the level you're aiming for, there was quite a wide confidence interval in this estimate. So I'm looking forward to seeing further research on this. There's a concept that PrEP is maybe medicalising sex too much, and so maybe it's easier to turn that round the other way and to say that this isn't an intervention that we're trying to design to do away with universal condom use. It's not something that we're trying to develop for daily medication for the rest of your life. It's an option, which means that should really be a person's choice. If this fits their choice for how to reduce their risk for catching HIV. You're looking really for an intervention that will help someone get through a difficult period or through a period in their life at least, where they will come through it without HIV being a complication for the rest of their life. So I like this slide from IPEX. Each of the rows represents the behaviour data from one of the people who seroconverted in the IPREX study, and the red blocks indicate a 3-month period where they would broadly be defined as at risk, and the cream blocks are when they were not at risk. And so you get a picture really that there's a huge interpatient variability between people in this study, but also an individual person changes their risk through the year and through the follow-up in the study. As a group, IPREX were incredibly well defined as a high-risk group. They were broadly young gay men, transgender women, who they had significant partner numbers. There was quite a lot of transactional sex. There was very low discussion of HIV prior to sex. There was very low condom use. There was high alcohol use. So it sort of overlaps with a high-risk group. But within that group, individual behaviour changes quite significantly, and individual risk. There's discussions about efficacy and effectiveness, and for me efficacy is whether an intervention works if you take it. And efficacy is quite often, it's what happens on a population level, but we can often make mistakes as community and as researchers when we enrol people into studies. So a highly effective intervention may not show effectiveness if it's used in a population that are not at risk in the first place, or if there's low use in a population that's at high risk. So sort of as an example, I guess, is the Bangkok tenofovir study, and this is an important study for finding the right conclusion that injection drug users should be able to access PrEP. At the end of the study, they found a clear efficacy result that showed there was benefit compared to placebo. This was in about 2,500 injection drug users in Thailand, and the end of the study included a very high-profile press release from the CDC recommending that drug users should be able to access PrEP. But over the study, only sort of 16 infections were affected. So that's a huge intervention. So most people weren't really at risk during the study. And at 3 years, there was no difference between the two arms. That's clearly not a mechanism problem. It's related to the fact that the incidence of HIV was very low, and it must have been quite a difficult decision for the DSMB to continue this study, and luckily they found a continued benefit. Like all the studies I'm talking about, these were publicly funded studies. So a problem really is how we match need to the intervention. And although several guidelines have raised the profile of PrEP, and I think they've done this for good political reasons, to make the entry criteria as wide as possible, the CDC and the WHO guidelines, in terms of individual relationships, when you're talking to someone about whether PrEP might be appropriate for them, it would be more helpful to start thinking about situation-based risk. So whether someone's had changes in their life, if they've been in a relationship and that's changed, they will be behaving differently. They may be more vulnerable when they're going back out to meet new partners. Sexual history, all sorts of aspects of sexual history are likely to predict or highlight people who could benefit from PrEP. STI history, recent STI history would be one of these, but if someone's been sexually abused, if you're aware of patients that's had complicated early life, that's a key predictor for becoming HIV positive later in life. Recent use of PrEP. In London, several people use PrEP 3 or 4 times during the year. Clearly, they would be much better. Their health would be much better served if they were offered daily PrEP rather than serial PEP. The IPREC study suggested just asking whether someone had recently unprotected anal sex without a condom would pick up many people. So cost effectiveness, I was going to put in a slide explaining cost effectiveness research, but that's far too complicated to be able to explain. It's all in the end driven by cost. And as an indication of where cost might go, the Clinton ceiling price for tenofovir for a year is about $50 for a daily dose. And I think certainly in Western markets that could have a really significant commercial increase in price and still be commercially viable, even if you multiplied that 5-fold. If PrEP was $300 a year, that becomes affordable on an individual level. Condoms and language, I guess. The optimal use of PrEP will not be with condoms. The optimal use of PrEP will not be in different partnerships where the positive person has got an undetectable viral load. There may be really good reasons for the negative partner to use PrEP in terms of reducing anxiety and understanding, coming to terms with risk in a different way. But I think recommending both routinely is not going to be very helpful. There's very good reasons why people don't use condoms, although they're not always very easy to explain. And all the PrEP studies so far have shown that risk compensation has not been a factor. The STIs are clearly important, but the primary aim really is to reduce HIV incidence. There's very interesting issues about quality of life. For 3 decades, the impact of fear in communities that are most severely affected by HIV is quite difficult to measure, perhaps until you take that level of fear away. So it would be interesting to see whether researchers can start to pull this out. I think that both treatment prevention and PrEP have the potential to normalise HIV, and stigma still remains high. But motivation can still include greater intimacy and control over your HIV risk. So this is a US doctor who at 60 decided that he was tired of the anxiety for worrying about if the condom broke or if he didn't use a condom. And although he says he's continuing to use condoms now, he noted that the reduction in anxiety was impressive. There's important safety concerns. We're using a medication. HIV testing is clearly essential, and monitoring. There's a potential for acute infections. There's an awareness of interactions with tenofovir especially. There's one of the US groups, the community groups, has pointed out that PrEP mustn't become an excuse to pressurise some individuals not to use condoms. This is a choice, should be an individual choice. And we need to maintain good monitoring on other STIs. And the issue of adherence, I guess, I put in a few slides. I don't know if it makes sense to non-English members of the audience, but there's an expression called TTFN, which means Tata for now. That could easily be Tuesday, Thursday, Friday, and now, if we find that the 4-dose PrEP is effective. Or if it's the mid-dose that people have trouble with, you could call it Truvada Tuesday. I'm not in PR marketing, you can tell. But something like this, we need to get the people that do marketing to get their heads around PrEP. Long-acting formulations have got the potential to overcome the adherence problems if they're successful. Ongoing research, I'm really keen on finding out what happened in PROUD and IPERGAY. There's other compounds that are being looked at. There's other preparations, so PrEP doesn't necessarily have to be a pill. Conclusions, I think I've already covered most of these points. EU regulatory block, or the bottleneck for why PrEP hasn't been submitted or approved or decided on in Europe, it's not really sustainable anymore in the light of the effectiveness data that we're getting from European studies. A challenge, I guess, is marketing in the community, and so I thought I'd end with a few slides that were given to me by a US activist, and if I just run through these, they hopefully show PrEP in like a more human light. And I'd like to thank our friends, colleagues and researchers who helped me with the talk, and apologise to any researchers if I mangled the data.
So we will now begin the discussion. Remember that if you want to ask a question, you can come to the microphone. You can alternatively go to www.hivglasgow.org/question to ask your question, and we will relay those questions. I think microphone one.
Mark Weinberg from McGill University in Montreal. Thank you both for these excellent talks. My question or two are actually to Simon, and the first question is that TAF is moving ahead very, very quickly and is likely to be approved during some time during 2015. Until now, the FDA, that as an example, has approved Truvada as PrEP, might consider requiring approval of a TAF-containing PrEP regimen. And so in view of the fact that TAF seems to have somewhat fewer toxicity problems associated with it than does tenofovir, what would be your perspective on whether the FDA should require trials using TAF rather than tenofovir to be required as part of an approval process?
I think TAF sounds like an interesting drug. Like regulatory perspective, I think this is all driven by independent researchers. I think it's unlikely to be of interest from the companies developing these drugs. So I don't think the structure for the FDA can require a company to do that. I'd like to see better PrEP. I'd like to see if TAF has the option of being better and it has advantages, then I'd like to see the data on that. I think it will come from independent researchers and it will come from private funding and public funding. And if there's a public need, then there should be public funding to see this work.
And a second question relates to the fact that in the United States, principally, there have been a number of groups that have actually articulated against the use of PrEP, arguing that PrEP may in some cases deprive people in need of therapy of these very same drugs. So how do you react to that?
I think the arguments within the community, or the difference of opinion within the community, I think has changed quite significantly even over the last year. So that whereas there was some sort of level of polarity, there was two different approaches, I think there's been much more of a consensus that as a choice, the efficacy data for PrEP means that it's an option that you shouldn't be denying people. And the cost, I mean, I forgot to refer to the manufacturing cost of PrEP. Some of these drugs will be off license and they'll be off patent, and it becomes incredibly cheap or inexpensive. And I think that even in resource limited settings, the PrEP is probably highly cost effective.
Andrew?
So, Simon, you had a really interesting slide about the condom use and the messaging. And clearly this is going to be very important. And I think that, we're all thinking what the situation is going to be for, let's say, a man who is essentially 100% using condoms so far successfully, uninfected, who's potentially going to stop using condoms and start using PrEP. We don't, and I don't think we ever will have, randomised trial evidence to actually be able to compare those two strategies, if you like. But it's going to, do you have thoughts about what the kind of messaging should be around that? Because I think there need to be explicit statements about how highly or not recommended that strategy is, and of course that is also going to have implications for the cost effectiveness.
I think PrEP is most effective in people who are at high risk, and so on that level, the public intervention really is focused on being able to identify people who are at highest risk in order to make this an option they might be interested in. So someone who's consistently using condoms and is quite happy using condoms, I can't see why they would want to change. But they might want to change. They might want to look at the data. They might want to reconsider really what their quality of life is using condoms. You know, what their relationship status is, how they interact with their partners. I think there's such a level of fear that really the level of fear has driven our sexual lives. And I think that's a shame. It's been difficult to measure. I think HIV is generally a difficult virus to catch. And so you will have the group of people who, without realising it, have obsessed their lives over the risk of HIV. And that's part of the reason they've stayed negative. But they've also probably stayed negative because HIV is quite a tricky virus to catch. And it's difficult with public health when the ultimate decision will be private. But I think that the people should be well informed and have the data.
Okay. Thanks.
Yes, José Gatel from Barcelona. The question is related. Do you know, or can you speculate what it's, or can you guess what it's going to be the future position of the EMA, the European Medicines Agency, respect to PrEP? Are they going to move? Are they going to request a dossier for approval, or what's going to be the position of the Medicines Agency in the near future? Because I think this is absolutely crucial if we like to have PrEP in Europe. Otherwise, we'll be able only to use it in the setting of clinical trials or in the setting of compassionate use.
I don't understand the way the EMA works. I think given the randomised data from a randomised clinical trial, the results are the same irrespective of geographic location. I do not understand why the EMA were not more proactive. And I think raising the profile now makes it difficult for them to continue not to be interested in this as an option. It's a real concern for public health systems to be using meds off-label without this indication. Having said that, PEP is widely used with absolutely no indication. All the ARVs are widely used, so there's no block that should block growing access to PEP, but clearly without that indication it's more complicated. I think the EMA needs to hear from you, needs to hear from other people in the audience, needs to hear from the community and the medical community, and Gilead have to be prompted to not accept no for an answer. I don't understand where this bottleneck is. It's just not but it's not acceptable, really.
And if I can comment on this one, I think the ECDC was quite cautious about it, requesting more data and data in the European zone. But when the result of both hypergay and PrEP will be available, which is early next year, I think part of this request are going to be fulfilled. So hopefully this will make a move possible in Europe.
Hi, Simon. Nice presentation. I'm a clinician in Canada. We've talked before, and I support PrEP, but the views that I have and you are certainly pretty far apart on the spectrum. And part of that is because nowhere do I see any mention about potential of developing resistance if you do acquire HIV infection, and how we're going to manage those patients who possibly get tenofovir resistance on PrEP. And the second thing is, nowhere do we see any discussion about acquisition of hepatitis C, which PrEP offers no protection for, and we're talking about making PrEP available for a very low cost for patients to take. When patients acquire hepatitis C and acquire hepatitis C repeatedly in the next few years, are we going to ask the drug companies to also give them a really low price for their hepatitis C treatment on more than one occasion?
Okay, so let's get this in perspective. You are talking about different populations out there whose level of risk is so high that your concern really should be compared to catching HIV. So compared to your lifetime use of ARVs, which actually I am doing pretty well on, and I think the drugs are amazingly more tolerable and more effective than we had in the 90s. And so the contrast shouldn't really, or the comparison shouldn't be between a potential toxicity from using an intervention that you've chosen to use instead of condoms compared to not taking this drug. It should be compared to the implications if you become HIV positive. You're looking at if you look at the populations that have been enrolled in PrEP and in IPGay, these are very high risk. These are people whose lives are not so far being able to exclude the risk that they could catch HIV. And in my experience, nobody goes out to catch HIV. It's complicated social factors and personal history that just means it's a result down the line of not having sufficient support, I guess, in order to be able to make decisions that keep your sexual health free of HIV. And I think the data from resistance is incredibly low. Data from tenofovir resistance. If you get tenofovir resistance, you use another drug and you become HIV positive. I think there's a poster here that's quite interesting. So there was 2 cases of people who were on tenofovir for hepatitis treatment. I don't understand why they weren't on Truvada. But on that daily, that wasn't sufficient to prevent infection. So they were effectively on tenofovir, PrEP and PEP, and they still became infected. So there's still lots of things we have to find out about. It's not 100% effective, but it's certainly much more effective than condom use, where most gay men use condoms most of the time, but some of the time they don't, and life is messy.
Okay, you didn't mention about the hepatitis C, but also...
Hepatitis C is just an STI. It may be a more serious STI, but it's still an STI that compared to over 3,000 gay men were diagnosed HIV positive in the UK. And I can't remember what the figures are in Europe, 100,000 in Europe or 2 million globally. This is where we should be looking in order to get the 90-90-90 targets that Stefano was talking about.
Okay, but I think you're also minimizing how difficult it is to treat patients with resistant virus.
Yeah.
I think tenofovir virus, well, perhaps somebody else would like to answer.
I'm not sure you're going to reconcile your point of view. Microphone 3.
Yeah, I will. So I'm Ian Rees from London. I acknowledge that sort of concerns around resistance and hepatitis C are worth considering, but when you look at the evidence, actually, sexual acquisition of hepatitis C in HIV-negative gay men is extremely rare. Although the PROUD study has reported a higher rate of hep C at baseline in the gay men going into the PROUD study, and that may well be because they have other risk behaviours such as injection and other types of drug use. But I think it is also worth continuing to monitor. In terms of resistance, I think there are a few people that seroconvert at the very beginning of studies of both IPREX and PROUD, and generally they have 184V, which is not a disaster in terms of management. They're a small number, and when people are highly adherent, they don't seem to get HIV, so the risk of resistance is not that high. My comment was going to be actually around this issue around condoms. I'm less concerned about condom displacement as well as Simon, but I do think we need to keep an eye out for the full results of the studies that are being conducted now, particularly thinking about that behavioural data and also the qualitative data. The PROUD study, I think, shows that the men that were enrolled were generally not using condoms because of perceptions around decreased pleasure or because their partners they felt were negative. So I think that people who are consistently using condoms are doing it on that basis of risk versus benefit, with benefit being, kind of protection and risk being loss of pleasure in sex. And if you've already made the decision that actually you prefer to have sex without condoms, then that's the group that we're looking for.
I was going to say, I think PrEP is really exciting, but the proportion of, but I'm more excited by treatment as prevention. So I think it will have a period and it will help us get to the 90-90-90, but the data on transmission for someone who has an undetectable viral load, and again you have all these issues of STIs, I think it would be nice to be talking about both issues together. But thank you for sort of confirming that behavioural change doesn't seem to happen just because condoms might not be being used.
So we haven't talked much about TASP. And Dominique, you have a question for Stefano?
Yeah. I think when thinking to the cascade 1990. I think if you don't enter the system, there's no way you're going to have a controlled viral load. So don't you think the focus should be on diagnosis? And in fact, in no country of the world we're doing so good for that, because there's a lot of late presenters as well in Europe and the United States. How do you think we can manage for that? Because
yeah, that's a very good question. In fact, I tried to point out that these first 90 was maybe the most difficult. They are all difficult, let's say. Yeah. But the first is really dramatic because the majority of infection, and that's true also for PrEP, because if everybody's on treatment and undetectable, then of course that's less need for if everybody would have been fully suppressed in the world with HIV. Mm. So the first is really a problem. How can we get this out, these people out? I think there are too many issues. One is social, and it's stigma and discrimination and the issue to put ourselves forward and be tested. And then we need to facilitate testing. So there is a technical part, there is a social part. I'm really not. We have still nearly 40% of people that is unaware. So I don't know how we can. But of course there are technicalities like the home-based testing should be. And we need also to make HIV, to normalize HIV in a sense, explaining that it's better that you are tested because then. So really, I'm not an expert on testing, but I think there are different issues. One is scientific, the other is social. I don't know how we will ever get to this 90, and if we don't get to this, then the cascade will definitely be less than 73% fully suppressed. So unfortunately I have no full answer.
Microphone 2?
Yeah, I think on the fact that treatment as prevention is obviously going to take maybe decades to become fully effective, that PrEP is additional armoury against the HIV infection for us to use in the interim. But my question is to Simon, that disappointing data from the States showing the lack of awareness and lack of uptake, I think it was 2013. Do you have any more up-to-date data, or has there been any educational campaign in the States to make MSM aware of PrEP?
So the problem with awareness studies is they'll always be out of date by the time you see them. And as I understand it, in the US, just like in many other countries, there's a lot of community and public health activity to start making it much more widely known that PrEP can be effective. So yes, I think there's been a huge change over the last, even over the last year. So I think awareness is now high. Most major newspapers have run studies on have run articles on PrEP, and most community organisations are involved in educational activity.
And we're seeing an uptake, an increase in uptake from that.
I think so. There's a paper here actually on the US uptake. It's not dramatic compared to treatment. I think the paper said about 500 people were started on PrEP in the first quarter of this year, which is tiny numbers really. But the US is a complicated healthcare system. So I don't think PrEP will ever be something that everybody wants to use. I think it will be something that some people want to use, and as there's more experience with it, I think I would hope that it would be used more.
I think it's time to close the session. Thank you for the very good presentation, and thank you for your questions.